Hello and welcome to our Grand Rounds. My name is Paul Naji. This is our Johns Hopkins University Biomedical Informatics and Data Science Grand Rounds that we host each month. It is really a pleasure this month to have one of our own colleagues, Dr. Blake Dewey, be our Grand Rounds presenter. Blake is a faculty member in our biomedical informatics section, and he serves as an assistant professor of neurology here, and had got his PhD at the Johns Hopkins Whiting School of Engineering in 2021 in electrical engineering under Dr. Jerry Prince. His doctoral research was about employing deep learning techniques to address data quality and harmonization challenges in extensive clinically acquired imaging data sets. After earning his degree, he undertook a research fellowship in neuroimmunology at the Johns Hopkins Multiple Sclerosis Center before becoming part of the neurology faculty in 2024. His research centers on the practical difficulties of utilizing clinically acquired images for quantitative analysis and integrating imaging results into precision medicine approaches. He teaches for us our deep learning with medical imaging and imaging informatics course here in the Q4. He also co-leads with me our Imaging Informatics Open Research Laboratory, where we are hosting many projects on imaging informatics. And he's also the lead across Johns Hopkins Medicine for our imaging architecture for our precision medicine initiative. So he's heavily involved with getting access to clinical images and providing them to researchers and trying to help us be able to use those for deep learning research purposes. Blake, it is a great pleasure to have you here today.
Thanks, Paul. And I have what should be a nice high-level talk for everyone today. I have nothing to disclose with regards to the contents of this talk. And today we're going to be talking about practical issues, both with the medical image analysis at scale, but also data quality, and describing some potential solutions such as harmonization. And so the title I've chosen today is Medical Image Analysis at the Hospital Scale. This is both an untapped potential, but also something that we are not achieving yet. This is an extremely difficult problem that leverages pieces of a ton of different departments and infrastructure leads across a hospital. So we're going to talk a lot about this today. We have an outline to this talk. We're going to start with some introductions to medical imaging, just very brief, and about the analysis techniques that we talk about. We're going to talk about these problems, most of these problems, not all of them, of medical image analysis at scale. And then we're going to talk about existing and potential solutions, and hopefully we're going to finalize with a nice call to action for some untapped potential. So we'll start with our intro here. So medical imaging, it covers a number of different things, radiology images, MRI, CT, X-ray, also ophthalmic imaging, fundus imaging, optical coherence tomography, then also pathology, whole slide immunofluorescence, but also visual light imaging, photography, skin imaging, and dermatology, for example. We have some examples on the right that bounce around from all these different disciplines. We have an X-ray, an abdominal CT, a brain MRI, a fundus image, and a pathology slide. And these are all very different imaging modalities, but they're all classed under medical imaging. And they could be two-dimensional, they could be three-dimensional, they can be color, they can be black and white, they can be all of these different things, including even four-dimensional. We have some dynamic imaging that we do that includes time as an additional dimension. And so these things can be all the way down from megabytes or even maybe kilobytes, all the way up to terabytes of data, depending on the modality. These pathology slides, whole pathology slides, can be absolutely enormous, and MRI scans are growing more and more every day. So how do we do imaging in clinical practice? There's a device. That device produces images, whether they're MRI, CT, X-ray, it's a camera, it's an MRI machine, it's an X-ray machine, any of these things. It's going to send its images to a PACS, a picture archiving and communication system. This is where we hold our medical images. Those images will be sent, in the case of radiology images, to a radiologist, in terms of fundus images or OCT, maybe to an ophthalmologist, and they will be read. They will be evaluated for medical use, and a report will be generated that is sent back to the PACS. Those images and report are then sent to the ordering physician. The ordering physician can then use those images and the report to make their decisions. Now, this is a fairly straightforward process, but it tells us nothing about what researchers have to do about this clinical imaging. Well, that's because we're in this bucket down over here on the side, along with all the patients, all the repositories that might get this data. We're not a part of this workflow, and that's because we're not a part of clinical practice as research entities, right? But we want to be able to tap into this because there's so much data available to us, and it's very important data and very representative data normally. So how do images get around? DICOM is the Digital Imaging and Communications in Medicine standard. It is both an image file format and a communication standard. It contains a lot of rich metadata, and we're going to see a lot of DICOM metadata in the next couple of slides. But basically, it boils down to key value pairs. So you have a key, a tag. It describes what thing is being measured or described, and then you have the value. Now, the fun thing about DICOM is it's actually been around and it's pretty much in its current state since 1993. The current version is 3.0. It was released in 1993. It has near universal acceptance among vendors. This is unbelievable in terms of medical technology that enough vendors have kind of come on board to adopt a standard. And it's still being updated, but with mostly backward compatible changes. Now, the problems that I'll talk to you about are not about the DICOM standard, but how vendors have chosen to implement it, which is really where some of the informatics challenges come into DICOM and medical imaging. So let's look at a DICOM metadata header. It is verbose. There is a lot of data that is stored in a DICOM header. And this is normally stored alongside each 2D image. So if you have a 3D image, you're storing this data 100 to 200 times as well. So you can see there's lots of different things on the board right here. There are a lot of different data types on the board right here, and a lot of different kinds of data that we're storing in the DICOM header. It's not all about the images. Sometimes it's also about the patient, about the location the image was taken, and just about how the data is stored. This is an MR, is a header from an MR image. It's actually taken from a freely available data set called ADNI. It's an Alzheimer's disease MR data set. So we can see it's correctly classed as an MR image here. It was taken on a GE Medical Systems MRI scanner. In fact, a Cigna, as they call them Cignas. It is the more kind of fun thing about this is that GE Medical Systems is what it says here, but I've seen GE MRI scanners say GE here or GEMS. These are strings, and they are free strings in some cases. So we start to see where some of these challenges can come up when we're talking about data quality, when a manufacturer can't even agree across time how it wants to describe itself. One of many problems that we'll talk about. We can also see another free text string in here. This is the type of image that was acquired. This is not a standardized language that's used. It's not a standardized vocabulary. It's a free text, and it's actually entered at the point of scan. It can be repeated, like can be called from a set protocol, but it can also be edited. So if this scan has to be repeated, some technologists will actually append the word repeat to the end of this little string, and we suddenly have millions and millions of different kinds of strings that we have to figure out what they mean. And they might only mean one thing to one technologist at one location. And so standardization is really difficult here. Move on to an actual number. This is the slice thickness. It's measured normally in millimeters, but it doesn't have to be measured in millimeters. We can also see that my program here has also told me that it's actually a string value. For those of you that do things in data types or in computer science, it's not always fun to store your numbers as strings, but DICOM does. It stores them as the decimal string type, and so we have a decimal string that is reported there. Inversion time, it's a MR parameter. It's measured in milliseconds here. Interestingly enough, the GE system measures it in a different way than a Siemens system. That's why I picked this one out. The Siemens equivalent of this number would be about 800. So we've already now seen about 4 different examples of how data can be messy, even in a very regulated standard. So we'll go on and pick another one. I know for a fact that this image doesn't have an echo train length of 1, but here it's reported as 1, simply because it just wasn't implemented correctly. Finally, we have the magnetic field strength, which we should know. It should be known. The magnetic field strength is extremely important to an MR scanner. This one is 3 Tesla, which is correct. But the interesting thing is not what's reported here, but actually how DICOM defines it. So this is directly lifted from kind of the DICOM standard. It reports magnetic strength, field strength attribute as a optional value. I maybe should be conditional, but an optional value. It's represented as a decimal string, and it's given this description at the bottom, the nominal field strength of the MR magnet in Tesla, which is a unit of measure. Then it gives example values of, and I'm going to skip the first one, 1.5 and 3. Those are very normal Tesla values for an MRI magnet. 15,000 is not a normal Tesla measurement for an MRI scanner. It is an extremely large measurement that would be very, very difficult to make and possibly very dangerous. That measurement is in Gauss, which is a different measurement for the same, different unit for the same measurement, but here it breaks the definition, even though it's given in the example values here. So DICOM metadata, very useful, can potentially be a data quality issue, especially when you start moving to different vendors, different sites, different modalities. All of these are different and must be very carefully analyzed. Medical image analysis is very broad. It covers a lot of different imaging types that we've talked about, but it also covers a lot of different things. It can be radiomics, which is like kind of textures and shapes and these kind of analysis. It can be volumetrics, which is getting a kind of a quantitative measurement of the size of a tissue or an organ or something like this. It can be computer-aided diagnostics. This was kind of hallmarked when mammography came to the forefront. Computer-aided diagnosis is really taken off since then, is used in a lot of different places, but it's a very, very useful tool for medical image analysis to help a physician make their decision or diagnosis or prognosis by providing them additional information. We can see a couple of examples here. We have a CT that's been segmented out into a lot of different structures. Same for this brain MR over here. We have a CT image without any label overlays that was used for a radiomics project, and we can see a 3D reconstruction over here that was done from a 3D volume. Now, all of these have been pretty much taken over by deep learning and AI methods. As we transition from more traditional image analysis methods to these deep learning and AI methods, we have to be considerate of training bias and data siloing. Data quality comes into this in a huge part because when we train these algorithms, they often do not generalize too far out from their training set without some very intense data augmentation. And so we really need to be careful when we're applying deep learning algorithms to data that it hasn't particularly seen, especially when we talk about jumping in terms of data quality or data acquisition method, say even going from a research scan to a clinical scan, which we'll come to in a little bit. So I've already shown you some problems, but now I'm going to actually give them titles and describe them a little bit more, how we have problems with medical image analysis at scale. So first, how has medical imaging grown? I'm going to show you a couple of studies that have existed over time, and we're going to see how this growth has happened. These are all going to be number of subjects with brain MRI studies in these different data sets. So this is the International Consortium for Brain Mapping, poorly termed ICBM, which was in the late 90s, early 2000s, and had an N of 800. This is now eclipsed by the Alzheimer's Disease Neuroimaging Initiative, ADNI, which has 3,500 subjects, but actually has even more scans because they have a very rich longitudinal data set, way more than 10,000 scans now with these 3,500 subjects. The UK Biobank has come in now with over 83,000 subjects as a part of their brain imaging study. Now, five-ish thousand of them have repeat scans as well. We're starting to see some exponential growth. Um, Within our health systems cohort, um, we have over 300,000 subjects that have a brain MRI study. So this is the scale that we're talking about. Even the largest research scans, even the largest research data sets are still dwarfed by the amount of data that we can present from a hospital scale. For comparison, I did my very first conference abstract in 2012. It had an N of 4, um, which in imaging science is a lot more reasonable. But in this case, it's completely nothing compared to even these large data sets. So big data, it causes big problems, and we're going to see why. Um, Less than 10,000 images, I would say it's feasible for you or a small team to investigate each image, to find out if there is anything wrong with the image, it's the image that you want, it's the data quality that you want, it's curated. We can curate all of these images if it's less than 10,000. You or a small team, it might take you some time, but you could definitely get it done. If we have more than 10,000, we're now in the area of needing simple QA. We're just looking at the images, we're making sure it's generally what we want. We're using teamwork here. We're splitting up this data set. We are chunking down to exactly what we need to know and nothing more, because you need to start burning through these images and going through them very quickly. If you have more than 100,000 images, even a simple QA is going to take probably not days, more likely weeks, even for a team to go through these images and even do the most simple of quality assurance. Um, we're now looking in the realm of automated quality assurance being required, or an extremely long amount of time to look through these images. And with large data sets, our data variability explodes. We no longer have single scanners or curated data sets that are being prepared for us. We now have messy data sets. These might be from patients in the ER. These might be from a wide variety of scanners and departments and all of these other things. They might not even be diagnostic quality. All of that exists in our clinical imaging data set, and we need to be able to separate that to be able to use it for research. And even accessing this data at this scale takes time. And so we really need to think about infrastructure here to be able to tell us and get us to these images quickly and efficiently. Now, this is a policy problem. This is a legal problem and ethical problem, as well as a scientific problem, because medical images are PHI. They're private health information. The metadata can contain direct patient identifiers. It can also be considered identifiable from the images themselves. In some images, especially in ultrasound, we can actually have burned in PHI. Means they've literally included text in the actual image that cannot be extracted because it's been completely written into the image. In 3D head imaging, we can extract facial features that could be identifiable. In fundus images or in OCT, we regularly consider that potentially retinal features could be identifiable. Now, we can obfuscate that, normally at a cost, but we need to always be thinking about whether or not the data that we have should be considered PHI and whether the work that we're doing should be considered human subjects research. There are lots of policies involved here. It's not straightforward, but in general, you can normally figure out a way to get towards a de-identified state, or what's called a limited data set state, where you have a limited amount of identifiers that can't be directly identifying, but will require extra information. I was actually at a conference last week and this person really didn't feel that fundus imaging should be considered PHI because it's not the same as the traditional iris imaging that's used for biometrics, and it's certainly less identifiable than some more intense data that we deal with on a regular basis. But that was his opinion. I will leave it to lawyers and ethicists to decide what I need to do with my data. So I talked about how medical image access is a problem. I'm going to show you why. This is a screen grab from our lighter version of our PACS system. I'm pretty sure this is actually my brain. I'm pretty sure that's who I searched. Um, then you get this on a web page. It's accessible through Safe, the Safe desktop, and you can actually download an image here. You can actually click this little save button and it will download the whole DICOM exam for you. It takes about 30 seconds. So for each scan that I want, not each study really, that I want, I'd have to search for the individual study. I'd have to either by accession number or patient ID or any of these kind of identifying features. And I would have to select it. I'd have to go to this next page. I'd have to click the save button. I'd have to wait for it to save, download through my browser, and save it to where I want. This is not scalable at all. It takes forever. I hate doing it if I have to. So why is this the way it is? Well, access to PACS servers is highly restricted. It is just a security measure. The way DICOM works, there's a lot of trust involved in DICOM, and so security measures to prevent access to DICOM improperly is to restrict who can access it. So access to PACS systems is limited. The extraction of images takes time. You have to actually query that system. It has to return the images to you. It's also potentially a production system. It's used in clinical work, so you can't just hammer it with queries and expect everybody to be happy with you. So you have to limit yourself, or you have to use a backup, which we do here. And then it also requires interfacing with either radiology IT or some other equivalent organization to get the access, the permission, and understand what is required to be able to access these images programmatically so that you don't have to go through this little save button. This is all a long negotiation and process that has to be taken into account when you're thinking about getting your images. Now, I'll show you a little bit simpler way later on, but this is a problem. And another reason that it's a problem is because it's actually limited in its connection to the EHR. We saw this imaging workflow above, but what I didn't show before was the EHR workflow. So what actually happens is we have this first swing where the ordering physician puts the order into the EHR system. That patient info and scheduling is sent to the RIS, the radiological information system, and the scan is scheduled and sent, and the appropriate information is sent to the device. Then we start our image acquisition, right? Now, what you don't see is a connection here between the PACS system, where the images are kept, images and reports are kept, and the EHR system. Now, they are linked, but those links are not very robust. They're normally simple callouts to the PACS system that allow you to view the images and the reports. Sometimes reports can be imported. It's not extremely common, but it does happen more and more as more physicians are wanting to get deeper into their radiology reporting. So this is a very limited integration. We don't have a strong integration here. So when we're talking about PACS systems, they're normally not completely linked. And so you can have things like patient reconciliation not being completely done. The DICOM imaging data can sometimes be stale compared to the up-to-date EHR system. So we really have to be careful when we start merging this data together because we want to make sure that the correct information is being presented in our images as it is in our EHR system. This is really important for multimodal analysis. So I showed you before how DICOM can be a really tough one to nail down in terms of informatics because you can't provide a whole lot of trust in the data that you have. Now, I've removed the information to protect the guilty here. I blocked out the Scanning Institute and other such things. But I have this DICOM data here. It is listed out as a neurospine study. The body part examined is spine. But the requested imaging procedure, meaning what came in from the RIS system, is an MRI brain with and without IV contrast. Now, that seems really specific and not what was done. The chances that they didn't do what was ordered is low, so likely this is an information challenge, not a actual scanning challenge. But if we actually look at the image, well, actually it is a brain. In fact, it's an MRI brain without contrast. And so we can just see here that these other parts of the DICOM tag are not correct. Likely they were put in at the beginning of the study, which was actually a spine, and then later they put on the brain portion later on, and those things stuck around. But so far, all I can trust is what's sitting on the screen in front of me, the image. Everything else is suspect. I'm going to do another one. I'm going to give you a hint. I blocked out JHU here because I want to I want to let people know that this is where they're at. This is an MRI JHU research with Reed. It is body part examined here is an abdomen. The requested procedure here is again MRI research with Reed. So it's a research scan. We know this, and we're pretty sure it's an abdomen. It's not. It's another brain. We really want to be careful when we're doing things like this because if we are querying our PAC system looking for the MRI brains, modality brain or modality MR, body part examined brain, we won't get this image. Nor will our radiologists get it when they look for previous brain studies. This is a quality improvement problem, a quality assurance problem, but it happens every day. I mean, these kinds of things are selected at time of scan, and there's problems that happen with it all the time. So we have to be careful with what we trust, and we want to be able to make sure that the data that we're getting is high quality data, especially in observational studies where we're going wide with our data sets. This is something that's near and dear to my heart, which is data quality of the actual images. I've talked a lot about the metadata, about getting the images, all these kind of things, but the actual images matter too. In order to be able to use some of these newer techniques, some of these AI techniques, you need reasonable quality data, or you need some method to improve it. And we have two kind of categories here. I call it research quality or clinical quality. It's not a hit on clinical quality imaging. They have different goals. Research quality imaging is normally cares more about qualitative or quantitative examination, so tends to be higher resolution, tends to have an improved contrast. It also tends to take a much longer time, or perhaps a little bit longer time, depending, and the acquisitions tend to be much more consistent. That goal is for research. They are doing more to have research quality imaging. Clinical quality imaging tends to be lower resolution, it tends to be a simpler contrast, it does tend to be quicker, which means they're getting more people through, and the acquisitions can be completely varied. There's a wide range. I'm showing you here MRI. There's also a lot of things here that are applicable to things like CT as well. All of these things can be applicable to a number of different modalities, but it's very obvious in MRI because it's such a variable technique. It's a very flexible technique is what we like to say. Now, you might be saying, okay, Blake, well, these are probably 2D images. You're not really showing them off very well because you've put down here in the bottom center and right these very thick sliced images and you're showing them in an off angle. That's not fair. Okay, that's fine. I'll flip them around. So you can see that in these research quality images, we get these nice 3D images. They're very high resolution in all the different directions. And this one in the bottom left that did look very nice on this view is actually hiding some low resolution information when we flip it around. And this is not a knock on any kind of radiology because these are diagnostic scans. They can get what they need from these scans. Normally, it's a qualitative or semi-quantitative analysis, counting locations, presence, absence, these types of things are doable with these kind of images, and that's why they get them. They're also very used to it. Radiology has an inertia to it where we've been trained up to look at these images. Why would we want to look at new images and potentially have some kind of difference that we wouldn't be able to account for? So just this is not a one way or the other, except, well, you probably should push towards the research quality images. But this is just explaining that when you start at a research uh, tool, a tool that's been validated on research quality imaging, you are not going to have a fun time when you apply it to clinical quality imaging. They're very different. And this is known. When we look at AI tools, we know that they have limited robustness. We know that they suffer from training set bias. And we know that the training data is almost often from research studies because that is what's openly available. That's what's available to these people to train their algorithms on. And we know that there's limited validation in real clinical data because we haven't seen it come out yet. And so I pulled this directly from a GitHub repo for Fast Surfer, which if you're familiar with Free Surfer, it's a brain segmentation tool. This is the kind of AI version of it that's supposed to be, quote, faster, which is fine. But you're looking here and they say all pipeline parts and modules require good quality MRI images, preferably from a 3T MRI scanner. Well, that already knocks out a good number of MRI scanners right there. It expects similar image quality to FreeSurfer, and notwithstanding any specific limitations, the resolution should be anywhere between 1 millimeter and 0.7 millimeters isotropic, which means in every direction. This is not a quality that clinical imaging has. This is a quality that research imaging has. And so you will be very, very hard pressed to find clinical quality images outside of specific institutions that meet these requirements. Okay, I got to complain for a long time, and now I'm going to talk about a couple of solutions. Each of these solutions is either ready, almost ready, or not quite ready. So when you look in the top left, you'll see if it's ready to use today, available soon, in a prototyping or beta testing phase, or it's an active research or development project. So first thing, I'm going to start with a green one because we need some hope in this talk. Projecting imaging data specifically at Hopkins, we actually have an imaging projection system that allows us to get access to imaging data. It all starts with the PACS. We actually take a one-to-one copy of it and put it in what's called a vendor-neutral archive. That vendor-neutral archive contains the same DICOM information that the PACS does. There we go. We extract a limited amount of DICOM metadata from that VNA and put it into a SQL database, in particular one table that covers some very limited DICOM metadata. And then we take a projection engine and it does a query on this SQL database. That can be any of these limited DICOM headers that we do, things like study description, procedure code, these very kind of broad things. We can also do series description, I think, which is one of the ones like the free text ones that I pointed out before, modality, and then of course, patient ID. And that actually gets us back what's called the accession number. The accession number is a unique identifier for each scan that we can then use to query the VNA. We can do a DICOM query on that and actually produce the images and store them in Azure. Now, this is actually a pretty robust system, but it does have some limitations. It only has a high-level DICOM tags, these like kind of study and series level tags. It has limited queries for that reason. You can only pick a certain number of things that you want to query from the SQL database to get those accession numbers. The de-identification is limited. We're still working on that particular piece, and the access to these DICOM images is file-based. When we store them in Azure, they're compressed DICOM files, and so they have to be accessed through a blob storage, and that can be difficult sometimes. So we have image projection, but we're still working on it. Now, I talked a lot about how we can't trust anything. Well, I'd like to be able to trust something. So we've been working on this with two master's students here, Carrie and Prahlad, who are both working on different approaches to predicting things like body part and acquisition method from the image and the metadata. So when we talk about these like free strings or even these images, how can we actually take that and produce limited standardized information from it? So this is extremely useful for things like automatic image filtering. So if I have an image and I don't have time to look at it, but I do have a second to put it through a quick neural network and decide what it actually contains, that's great. I can do automatic QA then. And then in addition to this, some of the work that Prahlad's working on actually has applications to harmonization and synthetic generation. so we can actually generate images that look the way that we want them to. And with direct classification, we can just take, say, an X-ray here, and we can produce the body part that's there. It does. We don't need to rely on this metadata anymore. This network can tell us, hey, that has a hand and a wrist. I'm like, okay, great. Now I know that it has a hand and a wrist. And if it's a larger image, maybe it has 8 organs in it. Okay, great. That has all those organs. I can use that for those organ tests. This other shared embedding space has a little bit of a more interesting tone to it in terms of like being able to get towards the contrast and things that we're looking for what's actually in the image in terms of how it was acquired. So in that way, we're leveraging this dual encoder called CLIP to encode both DICOM data and image data. Now, we can produce all the data that we want, but if we can't get it into a standardized format, we're going to be lost, right? I can produce spreadsheet after spreadsheet, and you'll probably just be frustrated with me. But maybe we could put it back into a standardized format. So Jen's done some excellent work putting together an imaging extension for OMOP. So I now have mentioned the big word in the bids department, OMOP. We can all clap and be properly satisfied now. OMOP is a very large standardized data format for observational health, and it covers mainly EHR, cost, notes, things like this, but doesn't yet have a strong connection to imaging. So Jen has developed some extension tables and vocabulary to hold DICOM metadata, as well as analysis results in the OMOP common data model. And we can see how this links in here. For those that are familiar with some of the OMOP data tables, we have the procedure occurrence table that links into our new image occurrence table, which then breaks down into these image feature table rows, which are then linked back into the measurement table. So we're now actually producing procedure occurrences and measurements that have to do with the DICOM metadata and image findings that come from analysis of those images. We have a reference implementation that we're in progress of putting together. It is agnostic to analysis method. In fact, it also includes provenance, which we can kind of see in this table right here. It allows you to specify exactly what version of the analysis tool that you use so that it can be reproducible. And since we're going to talk about reproducibility just for a second, we can talk about how we do reproducible analysis. The way that I have been a proponent of this is to actually use what's called software containers and actually use DevOps strategies to actually make reproducible and traceable environments to do your analysis. This means you can ship your analysis code to a number of different locations, number of different users. They will all be using the exact same environment, the exact same code that you did on yours, and it's traceable. We can actually track provenance alongside of our results. This is an example of the YAML type provenance that we generate through Radifox, which is our radiological image file ontology extension. And through this, we can generate so much useful information to be able to reproduce this work. We can look at the software module that was run, information about the container all the way down to when it was built and who built it, and actually a complete hash of the container, which means we can know down to basically to the bit level if this is the same container that we have right now. We can also look into how it was run, inputs, the required outputs. We can check to see if the file hashes are the same. This, again, is looking at the bit level to see if files are the same. Can look at parameters that were involved. This is also then auditable, so we could recreate the steps if we need to. All right, talked a lot about the actual metadata, the analysis, putting it all into place, but we do need to talk about how we tackle image variation. So Sam's been working on this for a number of years now. This technique is called super resolution. I talked to you a little bit about how clinical imaging has kind of a wildly different resolution profile than some of the research imaging where our models are being trained. So super resolution has the goal of enhancing the resolution, in this case of clinically acquired images. So we take something like these two images at the top. These are the same brain that I've simulated a 4 mm and a 5 mm slice thickness. So we can see how these are now very choppy. We get this aliasing, which comes across as this kind of staircase looking artifact, which is just a little bit more severe in the 5 mm one. Sam's method, called eclair, is built off of a previous method called s'more. We like our sweets. And they are both zero-shot methods. And the important thing about a zero-shot method is there's no training data bias. You are actually training on the image that you're testing on. You're requiring no outside training data. And this is an improvement that Sam made when he was bringing it to eclair. And we've also got a real signal model for MR in particular that allows us to generate synthetic data and actually train the model all on the low resolution image. It's relatively fast. It takes less, he says, less now than 5 minutes on a GPU, and we can get the results that look on the right here. So in low resolution, we see this very choppy low resolution image. For free, we can interpolate this. We can use interpolation methods to kind of get us a higher digital resolution. But as we can see, those staircase artifacts are still hiding in there. We're definitely still very blurry. We're not seeing too much here. But after eclair, in this case, we have these very nice crisp images. Now we can see that the 4 mm result is a little bit better than the 5 mm. But when we compare it to what we were starting with, we're starting to see a lot more features that might be useful to analysis here. This is a problem that we deal with in MRI, but it's not unique to MRI. The problem of contrast or acquisition method, which we solve using harmonization. Liamre graduated from the same group that I did, and we worked together on this. And Savannah has taken over this in after Liamre graduated. And the goal here is to normalize contrast, in this case in MRI, where there's a huge variability. This was pioneered in our group by using what's called image-to-image translation. This is a deep learning strategy that takes an image, runs it through a neural network, and generates another image. In this case, we're looking to generate images that are consistent with a certain acquisition strategy. And the important part of our work is that no cross-site training data was required. Previously, it was thought, okay, if you wanted to do this analysis, you'd have to actually acquire what a brain looked like in all these different situations. We can actually do it without that in a kind of a unsupervised, self-supervised manner that allows us to do this. Now we can see these are the same brains. They have the same anatomy, but they now appear as if they were all acquired in the same way, which is a very neat little trick because now these images all appear to, say, a deep learning algorithm as if they had been acquired in the same method as their training data. So we've now kind of bridged the training data gap to try and put our images into the distribution that the network is used to. Now, say you can't do any of this, you might want to make your method more robust. This is an admirable effort. It's extremely difficult, but segmentation techniques in particular are starting to do this. It's harder for radiomic techniques, but segmentation techniques, it's a little bit easier, although it's still quite difficult. And what they're starting to do is use synthetic data and real world validation, which is what I called for in our problem statement, right? So this is fantastic news. And in this case, the synthetic training data that they use is pretty terrible, right? They're generating these really, really poor images of all different kinds of acquisition methods, all kinds of different data qualities. This is the SynthSeg algorithm, which is starting to replace the FreeSurfer algorithm from the same group. But as you can see, it's new. This was just published in 2023, which might not be new in the days of LLMs, but is new for medical imaging. And we can see here that the test segmentations that they're doing are quite impressive. But we do leave a little bit to be desired in some ways. So we're happy with this movement, but there's still a lot to be done. So likely we'll want to include both robust training methods and some kind of harmonization, variation reduction techniques to bridge that gap completely. And one way that we might do that is actually generating realistic synthetic data. So when you go the whole way into synthetic data, it doesn't really look like anything. You don't get the benefits of some deep learning, which is to really drill down and get at specific features. So Sam and I have been working on this. This is looking to generative models to create realistic synthetic data. This is an example from our model. This was created purely from random noise. It's a full 3D image that was created through what's called a DDPM, a denoising network, a iterative denoising network, um, to create this realistic looking MR image. The problems that come into this is that it requires an absolutely massive amount of training data. When I said that it's tough to look through a lot of images, it is. We looked through about 80,000 MRI volumes to get us down to 75,000 MRI volumes finally. And those were relatively curated data sets. So it takes a lot of data and it takes a lot of time to prepare that data. And then additionally, I mean, this model took a number of days to train over many GPUs. So it's a very power and GPU hungry methodology, but we can produce images that are realistic, and that might improve how we can uh, work through some of these problems. All right, talked about problems, I've talked about solutions. Let's just finish it up by talking about potential. The potential that we're looking for is precision imaging. This is what I call imaging's introduction into precision medicine. We want to be able to create real, reliable clinical imaging feedback tools applicable to individual patients. This is a commercially available product that I have on the right, but it requires extremely strict quality control. You basically have to run their sequences and you have to do it right, and it has to be no motion and all of these kind of things to be able to do this work. So we're very happy that this work exists, but we want to bring it up to the level where we can actually use it in clinical practice. This can be used in clinical practice, but with the caveats. So to do that, we need to reduce measurement variability, we need to be robust against image variation, and we need to provide meaningful outcomes, which this is starting to do. We're starting to see these basically human growth chart type evolutions where we're able to track individual patients over time. So we have here, in this case, it's MS that they're doing. It's lesions, and they've also tracked brain volume. We also want to empower multimodal research. This includes deeper phenotyping. So we have kind of a classical example of Alzheimer's disease phenotyping, but we definitely want to include things like whole brain volume, cortical thickness, hypothalamus volume, FDG, tau, amyloid PET results. All of these things we want to be including in the phenotyping to represent the true holistic nature of the patient profile. This means fusing our imaging features with diagnostics, lab results, medications, but it does empower us to do cohort studies in large disease models using both EHR and clinical imaging data together. So this is part of the goal as well. And then we also want to create network studies. We want to use clinical imaging data that exists because it is frequently part of the standard of care. What we talked about in this talk is what we need for scalable analysis and for network studies. Scalable analysis is a crucial part of network studies because you need to be able to distribute analysis code. You need to be able to be confident that you're getting the right images. All of these things are all things that need to go into a network study. But that allows us to do this powerful federated analysis, and we can actually break down some of these data silos by doing these network studies if we can be confident in the images that we have. So what can you do? I apologize, that came out scarier than I was anticipating, this image over the site, but it's pretty impactful to me. We want you to push for high quality modern imaging when possible. If you're an ordering physician, hey, you should ask about what your imaging options are. You want to get to know your data. You want to know what types of imaging you have, the number of patients, the number of scans, all these kind of things that you have that you need. You want to be familiar with your IT and your research infrastructure so you can get through all of these bottlenecks that we've been talking about. And then we want you to reach out to your collaborators and to us because we want you to be part of our imaging network study. So I'll finish up with some thanks and acknowledgments from the Department of Neurology, from BIDS, as well as from the Whiting School at the Electrical and Computer Engineering Department. And this work is supported by a number of different grants across federal and private funds. And I'll end up here with questions.
Great. Thank you so much, Blake. Let's see. I do have a question about analyzing ultrasound images to enhance. Now, I think ultrasound is, yeah, ultrasound is always nice because it's non-invasive as a modality, but it really requires a lot of skilled technicians for trying to find the organ of interest. It's often very hard to interpret ultrasound images, but it does seem like how do you see ultrasound playing a role for in the research realm?
Yeah, I got to tell you, I run away from ultrasound projects. It's a very difficult technique that has an extreme amount of physical variability because of the viewing angle and of the amount of variability that you get from technologist to technologist. You really have a tough problem there. It's not unsolvable, but it definitely is something that you add on to all of these other things that come from vendors and image acquisition techniques and all these other things. So there is quite a lot of variability there. It's not easy, but there are starting to be people that are working on ultrasound from a quantitative aspect.
Yeah, the other nice thing about yeah, I do feel like ultrasound is an interesting one because it's a lot like sonar. So yeah, it looks for different reflections. It's very good for things like Doppler shift for looking at the physiology of vessels, but it can be very hard looking at structural objects as an imaging modality. So I guess what I think is interesting is I always like, everyone loves to talk about large language models today. But they're really very new, which is great. They're just a couple of years old. We're still seeing, the imaging revolution with deep learning started in 2012. So we've had over 10 years where we've had this new disruptive technology that all of a sudden we can get away from, before we had deep learning, we did feature engineering. We would do image processing, we would do skeletonization and region growing techniques to try to segment images. And now we have surrendered in some ways to deep learning techniques, just like today, just like the large language model folks do with clinical notes, where they would try to do feature engineering with named entity recognition and negation, and now they're trying to leverage large language models. But I think what's interesting is 80% of FDA applications are still medical imaging, so we're really still seeing a flood of new imaging algorithms coming in. At Hopkins, I think we only have a handful still for things like stroke protocoling, which is really where there's a timeliness aspect where we really want to get a read on the CT of the head to see if there's a bleed to make sure that we are responding correctly. We also use it for like mammography, for microcalcifications, and for screening purposes to make sure that it's very easy to have. It's really important to have a second read to improve your sensitivity and specificity of your reads. But I still see it's really very much fairly scattered within the imaging fields. And so you're saying you see a world where we are going to have a host of algorithms sitting in the clinical environment.
Yeah, I think so. And the issue that we come down in is where those sit in the clinical workflow, right? Because CAD has been around for a while. Computer-aided diagnosis has been around for a long time, especially in mammography, where it really took off, looking for things like microcalcifications, as you mentioned. But we're moving towards that way. But it's I caution people that push the question, oh, this is going to replace radiologists, because anybody that works with AI models knows that that's not true. They're first of all quite dangerous in some ways. They can be unfair, and they really are right now a part of the research suite bridging into the clinical suite. Because we really have, and I heard a couple of great talks. I was at Arvo last week, heard a couple of great talks about bringing your techniques into clinical realm, and three of those people were regulatory agents. It's a huge regulatory burden to bring software as a medical device, and for good reason. It has to be very heavily validated. There have to be very clear guidelines and guardrails established. And part of those guardrails and guidelines are to establish that you have tackled some of these problems. If your worry is image variation, you need to have strict guidelines on how the images need to be acquired to use your technique. That the commercial product that I showed, That's exactly what they did. They said, this is only valid for this one MRI scan. If you got something else, we cannot help you. Our results are invalid. And that's what the FDA made them say. You have to put these guidelines down because otherwise we cannot trust your product to be reliable.
But what's interesting is, I guess so you're right. I like this idea of a spectrum. When we talk about, and so we do lots of observational research, which is actually looking at data collected for clinical purposes and see if we can use it for research. I like this, and we see this nice analogy between clinical notes and imaging, where we're trying to learn from them, and can we look at the natural variation to see how people respond to treatment. But I'm interested in this aspect of, so you have clinical data, but often the AI algorithms are really designed for research data, and they're not designed for clinical data. And so when they get approval, they get deployed clinically, they have to force clinical changes to the workflow to make sure that they're getting higher quality data for them to be useful.
Ones that the clinical workflow is resistant to.
Yeah. And
You
the bias
basically have to
my
come
new
down
MR
from
scan.
a,
Yeah.
yeah, you have to come down from above basically to get these things implemented, which is problematic for some of the commercial aspects as well, because you're now limiting who can use your product, which they hate.
Yeah.
But the FDA is very adamant that these guidelines and guardrails be established because otherwise it would be an absolute mess. Um,
I do like that, I do, I think it's interesting how the DICOM, I like the idea, so the DICOM standard, so before we had modality work list, which is when, so right now, you're right, physicians enter in an order in an EHR, that order goes to radiology, that data gets populated into the DICOM header. So before, that actually didn't come until like 1996. And before that, a technologist would have to type in 40 fields into the CT and MR scanner to identify the patient. And the odds of them getting one of those fields wrong was around, I guess, around 15%, that there's a probability there's some sort of data quality error. So by moving to modality work list, they have to select the name off of a list, and then that kind of improved the quality, but it still only got us to like the next round. It seems like there's now like a data quality issue at more of the 2 or 3% range, and then you have a lot more issues with the variability in techniques that are localized to the site.
yeah, and I guess you're right. I should be thankful that we're not typing in patient information anymore.
I was there. Yeah, so
RIS did make a huge improvement there. But the problem with stale data is still there, right? The lack of syncing up between EHR reconciliation and the PACS system can cause some missteps and and can cause things. But yeah, we're really are now we're able to tune back down into, okay, how is this actually acquired versus do we actually have the right patient? Which
yeah, when you
that I
say
can be okay, fairly confident of.
Yeah, so when you say stale data, you mean so the images were acquired 5 years ago, and they sit for a long time. That's actually the medical record, just like the other pieces of the EHR. And so we are required to retain them for at least 7 years for adults and till the majority for minors. So up to 18 years for minors. And so we have a long retention policy for these images. And so what happens when their name changes or other demographics changes? Often those don't flow down to the image level and get changed at the DICOM tags. And so that's what you mean by stale data, is that
Yeah.
things have changed around the patient identifiers. Their medical record number might have changed, or their name might have changed, or some other field
Or it
around
might have
it.
been merged with another medical record number. I mean, merge
Yeah.
and split is a huge problem with reconciliation.
Yeah.
Um, Also, conflicting medical record numbers. This happens uncommonly, but you have multiple medical record numbers. We still maintain, I think, two at Hopkins, at least two. And yeah, it can be very complicated to make sure that you are querying the right data. And when it comes to ethics, right, you want to be very confident that you're querying the right data, right? When you're talking about programmatic access to a database that potentially includes all of the clinical data, um, you need to have measures in place to make sure you're querying the right data, you're collecting the right data for research.
I also like this idea that, or I kind of concur with this premise that some of the current AI clinical algorithms are kind of a half step. They didn't really, they force a different level of image quality at the site, so they kind of only can handle a very narrow window of data. And so what we really need to get to fully get to and tap into all of medical imaging, we need to have an intermediary where there's a data quality check that can actually look at the pixels and using deep learning, use it as a kind of a data quality agent to say, is this image going to blow up this AI algorithm, basically, in terms of what it's expecting to see.
Yeah, we kind of have a view as like a funnel, right? All the imaging data, and then the tiny little end of the funnel is what's actually acceptable to the algorithm. And you can just think of it like a clinical trial or a drug trial. These people, these drug trials and these clinical trials have full-time staff. They're just doing quality assurance. They're just ensuring that these things are right. And that's the standard by which we evaluate our medications and our medical practices. And so when we talk about clinical practice, we can't necessarily have that level, but we have to have something in there to ensure that the data that we're using
And
when we
these
don't have a human in the loop, right?
Yeah, these CT and MR scanners can easily cost over $1 million. So generally, they have to have a relatively long lifespan to be able to
Mhm.
recoup those costs. Well, great.
Exactly.
Thank you so much. We're out of time. Thank you so much for the grand rounds and a peek into the world of imaging informatics and how uh, quality and AI is going to try to actually add to start solving that gap in trying to get value out of these algorithms in clinical care. Thank you so much for attending today, and we'll reconvene in September for our next grand rounds. Thanks again, Blake.
Thank you.